Polysaccharides are large polymers and can be up to many thousands of units long. Polysaccharides are found free or bound to other cellular structures such as proteins. Polysaccharides are not sweet and in general they are not soluble in water. Like disaccharides, the monomeric units of polysaccharides are linked together by glycosidic bonds. The orientation of the glycosidic linkages in these polymers is different. At the same time, they are linear and branched macromolecules and as a result have different properties. Polysaccharides are a very diverse group of chemicals when it comes to their structure. Four of the most biologically important polysaccharides, starch, glycogen, cellulose, and chitin, are all composed of repetitive glucose units, although they differ in their structure. Polysaccharides contain slight modification of the repeating unit. Depending on the structure, these macromolecules can have distinct properties from their monosaccharide building blocks. Polysaccharides are an important class of biological polymers. Their function in living organisms is usually either structure or storage related. Starch and glycogen are nutritional polysaccharides. This group of polysaccharides is used exclusively for the storage of sugar molecules. They are easily broken down by the organism, making them allowing for the rapid release of sugar to meet rapidly changing energy needs. Starches are polysaccharides with molecular bonds between sugar molecules that human intestinal enzymes can break down. Plants are able to synthesize glucose, and the excess glucose beyond the plant's immediate energy needs is stored as starch in different plant parts, including roots and seeds. The starch in the seeds provides food for the embryo as it germinates. The starch that is consumed by humans is broken down by enzymes such as salivary amylases into smaller molecules such as maltose and glucose. The cell can then absorb the glucose. Starch is the stored form of glucose in plants and is made up of mixture of amylose and amylopectin. Starches are an important source of calories and can be found in foods like rice, potatoes, wheat, and corn. Starches don't taste sweet like simple sugars because they don't activate taste buds in the same way. Amylose has the simplest structure of any of the nutritional polysaccharides, being made up solely of glucose polymers linked only by alpha 1 glycosidic bond. Amylose is insoluble in water and is harder to digest than amylopectin. Amylose is produced in plants for energy storage, and since plants don't have rapidly changing demands for glucose, its compact structure and slow breakdown characteristics are consistent with plants' needs. More complicated polymers of glucose are possessed by amylopectin in plants and glycogen in animals. Both compounds contain long glucose chains with alpha 1,4 bonds like amylose, but unlike amylose, these long chains have branched of alpha 1,6 bonds. Amylopectin is the less branched of the two, having such bonds about every 25-30 glucose molecules, whereas glycogen has branches about every 18 to 12 glucose molecules. Branching plays important roles in increasing water solubility and in providing more ends to the polymer. In animals, glycogen is broken down starting at the ends, so more ends means more glucose can be released quickly. Again, plants, which have a lower need for quick release of glucose than animals, get by with less branching and fewer ends. Glycogen is analogous to starch and is sometimes referred to as animal starch, having a similar structure to amylopectin, but more exclusively branched and compact than starch. Glycogen is a polymer of alpha 1 4 glycosidic bonds linked with alpha 1 6 linked branches. Glycogen is found in the form of granules in the cytoplasm in many cell types and plays an important role in the glucose cycle. Glycogen forms an energy reserve but can be quickly mobilized to meet a sudden need for glucose, but one that is less compact and more immediately available as an energy reserve than lipids. If the glucose is not immediately needed for energy, the body can store up to 2000 calories of it in the liver and the skeletal muscles in the form of glycogen. Once glycogen stores are full, carbs are stored as fat. If you have insufficient carbohydrate intake or stores, the body will consume protein for fuel. This is problematic because the body needs protein to make muscles. Using protein instead of carbohydrates for fuel also puts stress on the kidneys, leading in the passage of painful byproducts in the urine. Glycogen serves as the secondary long-term energy storage in animals, with the primary energy stores being held in adipose fat tissue. Glycogen is made primarily by the liver and the muscles. The two major sites of glycogen storage are the liver and skeletal muscle. The concentration of glycogen is higher in the liver than muscle, but more glycogen is stored in the skeletal muscle overall because of its much greater mass. In the liver, glycogen can be composed of 8%, roughly 100 to 120 grams in an adult, of the fresh weight soon after a meal. Only the glycogen stored in the liver can be made accessible to other organs. Glycogen is also stored in the uterus and fallopian tubes and is an important source of glucose. Endometrial glycogen concentrations are correlated with fertility in humans, indicating that glycogen is an essential source of glucose during early pregnancy. Glycogen is not as energy rich as fatty acids. Why do animals store any energy as glycogen then? Why not convert all excess fuel into fatty acids? Well, glycogen is an important fuel reserve for several reasons. The controlled breakdown of glycogen and release of glucose increase the amount of glucose that is available between meals. Hence, glycogen serves as a buffer to maintain blood glucose levels. Glycogen's role in maintaining blood glucose levels is especially important because glucose is virtually the only fuel used by the brain except during prolonged starvation. Moreover, the glucose from glycogen is readily metabolized and is therefore a good source of energy for sudden strenuous activity. Unlike fatty acids, the released glucose can provide energy in the absence of oxygen and can thus supply energy for anaerobic activity. Have you ever had the experience of hopping on a new diet and losing 10 or more pounds in the first week? Maybe you have had pizza night with your friends, waiting yourself the next morning and find out that you are apparently gained 3 pounds from that one meal alone. What has really happened in both cases is that your body's water weight has fluctuated. When we talk about water weight, specifically in the context of dieting and fitness, what we are mostly talking about is water stored in the cells of the body, often joined to glycogen. Thus, even as it will feel good to shed 5 to 10 pounds simply from a few days of exercise mixed with a calorie restricted diet, the weight loss will be primarily from a reduction in glycogen stores and water. In other words, what you will have lost in the beginning is really little more than water weight. The dramatic water loss at the outset of a low carb diet is usually water weight. Your body stores carbs in glycogen in your muscles and liver. When your diet does not provide adequate carbohydrates, your body must call upon its reserves, meaning it releases the glycogen, which is bound up with a whole lot of water. As your body burns through the glycogen, the water is also released, meaning the initial success of a low-carb diet may not be what it seems. Thank you for watching.